student of School of Pharmaceutical Sciences. So today I'm going to present topic on drug discovery and development. So first of all, we are talk about what is drug discovery. So drug discovery and development, it is a complex and lengthy process that involve the identification, designing, testing of new medications, and it is typically consist of several stages, each with its specific goals and challenges. And here we uh, and here is a general overview of the drug discovery and development. So first is the introduction. In the past, most drugs have been discovered either by identifying the active ingredient from traditional remedies or by serpentitious discovery. But now we know diseases are uncontrolled at molecular and physiological level, and also shape of the molecule at atomic level is well understood. And what is the history of drug discovery? So in pre-1990s, herbal drugs, serpentitious drugs are discovered, and in 90s and 30s, vitamins and vaccines are discovered, and in 1940s, antibiotics era and R&D boost of WWT, and in 1950s, new technologies and discovery of DNA, and in 1960s, breakout breakthrough in etiology, and in 1970s rise of biotechnology and use of ITs, and in 1980s commercialization of drug discovery as well as combinatorial chemistry, and in 1990s robotics and automation discoveries. And in broader sense, drug discovery and development can be identified. It is a process start with identification of disease and therapeutic target of interest. It include methodology, assay development, lead identification, characterization in vitro, and here are different stages include like basic research, feasibility studies, program, non-clinical development, clinical development, and see the graph. And here is 10,000 compounds are discovered, but only 250 compounds are entered in preclinical data, and it takes 6 to 5 years, 6.5 years. And after that, only 5 compounds out of 250 compounds are entered in clinical trials, and it takes 7 years. And in only one compound from 5 compounds will be FDA approved, and it takes 1.5 years. And now the process. So first is a target selection, and second is a lead discovery, medicinal chemistry, in vitro studies, preclinical studies, and clinical trials and therapeutic. So first we are talk about, first we select the target, then we go with its lead discovery. We discovered the lead, and then we go with medicinal chemistry. We discuss the medicinal data, medicinal chemistry, and after that when it passed from this, and it will go with in vitro studies, and after that preclinical studies, and then in clinical trials. So first is target selection, in which cellular and genetic targets, genomics, pre-atomics, bioinformatics. So bioinformatics, there is a huge scope. And in lead discovery, first is a synthesis and isolation, and in combinatorial chemistry, and then assay development and high throughput screening, also called HTS. And in medicinal chemistry, library development, SAR studies. SAR is basically structure activity relationship. And after that, in silico screening and chemical synthesis. And in vitro studies, we check the drug affinity and selectivity of the drug. And after that, cell disease models and what its mechanism of action, lead candidate refinement. And in preclinical studies, animals model of disease, behavioral studies, functional imaging, ex vivo studies. And after that, when it clear all the uh, stages, it pass to the clinical trials and check the therapeutic value of the drug. So first of all, we talk about drug discovery. So drug discovery, it is a phase during which the candidate or target of interest are selected on the basis of their pharmacological basis. So drug discovery includes random screening, molecular designing, drug metabolite, drug metabolites, and serendipity. And first is our target selection. So target selection has cellular and genetic targets, genomic, pre-atomics, bioinformatics. So target selection in drug discovery is defined as the decision to focus on a finding an agent with a particular biological action, and in which target identification we identify the molecular targets it involve in disease progression, and target validation to prove the manipulating the molecular target can provide therapeutic benefit for patients. And next is the cellular and genetic targets. It involves the identification of the function of a potential therapeutic drug target and its role in the disease process. After that, genomics. It is a study of genes and their functions. So basically, genomes aim to understand the structure of genome. It includes the mapping genes and sequencing the DNA. And proteomics, it is a study of proteome, the complete set of protein produced by the species using the technology of large scale protein separation and identification. After that, bioinformatics. So bioinformatics, there is a huge scope of bioinformatics. Bioinformatics is a branch of molecular biology. It involves the extensive analysis of biological data using computers for the purpose of enhancing biological research, and it play a key role in various stages like target identification, computer screening of chemical compounds, and after that lead discovery. So lead discovery has four types. Like first we go with synthesis and isolation, then combinatorial chemistry, assay development, and at last HTS, or also called as high throughput screening. So first of all, lead discovery. Lead compound, it is an organic molecule that acts as a prototype drug around which further optimization is centered or focused, and identification of small molecule modulators of protein function, the process of transforming these into high content lead series. So next is the synthesis and isolation. Separation of mixtures. First we separate the mixtures, then we separate the impurities, and in vitro chemical synthesis. After that, biosynthetic, we make a biosynthetic intermediate. So this is the synthesis and isolation. Then combinatorial chemistry, it is a rapid synthesis of computer simulation of large number of different but structurally related molecules in which search a new lead, and optimization of target affinity and selectivity, and as well as ADME properties. ADME basically it is adsorption, distribution, metabolism, and excretion properties are reduce toxicity and eliminate the side effects. So next is the assay development. It is used for the activity of a drug and discriminate between the compounds, as well as evaluate the express protein and enzyme substrate interactions. So at last, the high throughput screening. It is a process by which pharmaceutical companies are able to obtain initial screening data up to 1 million compound testing against as many as 50 different biological targets. The expansion of data collection by several orders of magnitude is primarily due to the advancement in robotics. So first is screening of drug target, identification of highly target specific compounds, and at last medicinal chemistry. So first of all, what is medicinal chemistry? Medicinal chemistry is a value of medical value of compound. So basically it is a branch of chemistry. It is a branch of organic chemistry. It is a discipline at the intersection of synthetic organic chemistry as well as pharmacology. Focuses on small organic molecules, not on biologics, as well as not on inorganic compounds. And it is used in drug discovery, hits, and lead optimization. It is hit to lead. After that, library development. So library development, it is a collection of stored chemicals along with associated database, assist in a high throughput screening, and it is help in screening the drug target hit compounds, and it is based on organic chemistry, not on inorganic chemistry. So next is the SAR studies. So SAR is basically structure activity relationship studies, in which it helps to identify the pharmacophore, and pharmacophore is a precise section of the molecule that is responsible for the biological activity. So it is enabled to prepare more active compound and allow elimination of excessive functionality. After that, in silico screening, it is a computer simulated screening of chemicals. It helps to find the structure are most likely to the bind to the target drug target, and it is economic than HTS. It means it is economic than high throughput screening. So next we are talk about chemical synthesis. Chemical synthesis, it involves production of lead compound in suitable quantity and quality to allow the large scale animal and eventual extensive human clinical trials. Optimization of chemical route for bulk industrial production and suitable drug information. And last, in vitro studies, also called in glass studies, because of it is studies using component of organisms like test tube experiment. That's why we called it is in glass studies. So examples are cell derived from multicellular organisms, subcellular components like ribosome, mitochondria, cellular subcellular extract like wheat gram, wheat germ, reticulates extract, and purified molecules, DNAs and RNAs. And it is a theoretical and practical aspects of drug development. So preclinical study, the aim of this stage to satisfy all the requirement. This pass all the tests like we select the target, we discover the lead, medicinal chemistry, and pharmacological testing, preliminary toxicology testing, pharmacokinetic studies like ADMEs, absorption, distribution, metabolism, excretion studies, and as well as chemical and pharmaceutical assessment to assess the feasibility of large scale synthesis and purification. After that, in which preclinical study, it is the experimentation using a whole living organism. It gives the information about what its metabolic profile, what its toxicology profile, and what are the drug-drug interactions. So animal model of disease state, test condition involve induced disease or injury similar to the human condition must be equivalent in mechanism of cause, and it can predict toxicity in 71% of the cases. For example, SCID, it is a severe combination immunodeficiency mice HIV. And next is a behavioral studies. It is tool to investigate the behavior result of the drug. It is used to observe the depression or mental disorders. So for example, like disappear based for saving tail suspension, reward based, anxiety based. Functional imaging, method of detecting or measuring changes in metabolism, blood flow, regional chemical composition, and absorption. And the tracers are used in functional imaging are MRI scan, CT scan. So next is the ex vivo studies. Experimentation on tissue in an artificial environment outside the organism with the minimum alteration of natural conditions, counter ethical issues, and for examples are measurement of tissue property, realistic of model for surgery. And after that, clinical trials. It is further divided into the four parts: phase 1, phase 2, phase 3, phase 4. So first, what is clinical trials? It is a set of procedure in medical research and drug development to study and safety and efficacy of new drug. It is essential to get marketing approval from regulatory authority and may require up to 7 years, and it takes up to 7 years. So first of all, we are talk about phase 1. Phase 1 is the clinical pharmacological evaluation. It is a first stage of testing in human subject. It takes 20 to 50 healthy volunteers, and it is concerned with human toxicity, tolerated dose range, and pharmacology dynamics. As well as we are talk about phase 2. So it is a controlled clinical evaluation. It takes 50 to 3000 individuals, and it control the single blind technique, and it concerned with safety, efficacy, drug toxicity, and drug interaction. Next we are talk about phase third. So phase third, it is also called extended clinical trials, and it is most expensive and time consuming, and it takes 250 to 1000 patients, and it called controlled double blinding technique, and it concerned with safety, efficacy, comparison, and package insert. Phase fourth, post marketing surveillance, designed to detect any rare or long term adverse effect. It is used for the monitoring of adverse drug reaction, and some are recently approved drugs by FDA. It is the data of 2023. So first is a lecanemab. It is companies are involved in ESA. But these all are the blockbuster drug of 2023. SRP 9001. The companies are involved in Sarepta Roche, and it is used in gene therapy. Next is intravitreal uh, peg, pegcetacoplan. It is companies involved in Epicillus, and it is used in C3 inhibitors. And donanemab, it is companies involved in Lilly, and it is used in Alzheimer disease. And next is the RSVP F3. The companies involved are GSK, and it is used for vaccine of older adults against respiratory symptoms, and it is approved by PDUFA. Thank you so much. Thank you for listening me. भाई कुछ नहीं हुआ 16 मिनट ये कैसे पॉसिबल है इतना सारा तो बोला है मैंने